Hello everyone. This week we're going to talk about the different physical and chemical control methods that we use to basically control the abundance and growth of microbes. Why is it important to talk about this topic? Well, because microbes, whether those microbes are viruses, fungi, bacteria, and microbes are ubiquitous. They're found everywhere. For example, they're found on objects that we handle every single day or touch every single day, like your tabletop, like your computer screen, your laptop, your books, pens and pencils, etc. The list can go on. These surfaces do have microbes living on them. It is important for us to keep those surfaces clean to basically try to prevent the spread of human disease. Now, there are physical methods that we use, such as, heat, high temperature, basically radiation, filtration, things like that. And then there are also chemical methods, we can use things like bleach and hydrogen peroxide, iodine. We can also use things like soap and detergent. So we're going to talk about all of these different methods. But again, the whole point of doing all of this is because we want to control, right, the abundance of these microbes. We don't want things to get out of hand, otherwise, we can get sick. So let's go ahead and start talking about these different types of methods that we use. Let's go ahead and talk about some different microbial control agents that are used. We'll start with chemicals that are known as germicide or microbicide. Now, these are chemical agents that kill microorganisms. Now, these agents, they would end in "-side". So when you see the suffix "-side" or "-sidal", by the way, now, that suffix means that these agents have the capability of killing the microbe. Now, the prefix that would come before that suffix "-side" would tell you which microbe is getting killed. So if I say bactericide, then this is an agent that kills bacteria. Fungicide would be an agent that kills fungi. Viricide, it kills viruses. Sporicide, it kills spores. Okay. So these are some, again, chemical agents that are used, and they go ahead and they kill, that specific microbe that are targeted. Now, there are also microbistatic agents, and those are chemical agents that would inhibit the growth of microorganisms. Not kill them, okay, but actually inhibit their growth. Kind of, you're putting them in, keeps the microbial population in check, okay? Now, such as, again, bacteriostatic, fungistatic. So again, the suffix would end in static, okay? Now, usually static treatments, okay, as I said, they don't kill that infectious, microbe, but they just go ahead and kind of, keep their growth under check. Now, those static chemical agents, they are usually less toxic to us humans, okay? Also when used, they can actually preserve the integrity of the item that you are treating. Because of their reduced toxicity, okay, sometimes what they do, okay, guys, is they can actually impregnate. So they actually like inject such static, chemicals into plastics to prevent the growth of microbes on plastic surfaces. So for example, they go ahead and they impregnate products that are usually used by kids, like, a lot of the kids' toys are made out of plastic, so they go ahead and they inject it into the plastic, within the toys, or they go ahead and they inject it, impregnate it into the cutting boards, for example, for food preparation, okay? And that's to kind of like, prevent the microbial growth, or to prevent the excess microbial growth on such surfaces. So this is gonna, one way of how they do it to, again, help prevent those, bacteria from multiplying, very fast. Now, of course, again, how can we determine whether a treatment, is a cidal treatment, that will kill the bacteria, for example, or whether it's a static treatment that will just inhibit its growth? Well, it kind of depends on so many things. It depends on the type of microorganism that you are targeting, because we're going to learn about how some microorganisms, they are pretty resistant, okay? And then we're going to talk about also the concentration of that chemical that you're using. Of course, is also going to make a difference how much of that chemical that you are using. But again, you have to think of, is that concentration going to be toxic, right? For example, like is that concentration toxic to us humans? And so it would be hard for us to handle such high percentage of certain chemicals. And then another thing is the nature of the treatment itself, like the chemical that you are using, for example, okay? And we're also going to talk about like some other than chemicals, there are other physical methods, okay, that we can use to, again, either kill the bacteria or kill the microbe in general. And there are also physical methods that are used to go ahead and also inhibit the growth of microbes. The different physical and chemical agents that are used to go ahead and either kill the microbe or inhibit its growth. Now, what do these agents target? They target one of the following. They either target the cell wall of the microbe, so they either block the synthesis of that cell wall. So if the microbe cannot make its cell wall, the microbe is going to be affected. Or they go ahead and these agents, they would digest the cell wall, so they would break it down. Alcohol, certain detergents, and certain chemicals can actually affect the cell wall of the microbe. Now, they can also target the cytoplasmic membrane, or basically the plasma membrane of the microbe. So these agents, what they would do is they would bind to the bilipid layer of the plasma membrane, and they would go ahead and kind of break it open. And so as the plasma membrane breaks open, the chemicals would enter into the cell, and again, affecting the cell. And certain detergents can do that. Other agents, what they do is they go ahead and they affect the cellular synthesis. So they affect and they interrupt the synthesis of certain proteins inside of the cell. They disrupt like the ribosomes that are needed to make those proteins. They inhibit the proteins, and so when they basically disrupt the whole process of making proteins, those proteins are important, for several reasons. Some proteins are needed to grow, for growth. Some proteins are needed to help the bacteria, for example, metabolize certain nutrients. And so without these proteins, an example of proteins are enzymes. So if you kind of stop the production of certain enzymes, then again, you're going to affect the microbe's growth rate. Things like formaldehyde, certain radiation. We're going to talk about different types of radiation out there. There are gamma rays, there are, for example, UV rays. So we're going to talk about the different radiation that can be used. And then what's another thing that can be affected? The proteins themselves. So some of these, agents that are used, they go ahead and they denature the proteins. They basically break down the proteins. And as I said, proteins are super important for many reasons, okay? Metabolism, growth, etc. As I said, enzymes are proteins. So again, you can be denaturing certain enzymes, and without those certain enzymes, the bacteria cannot go through certain processes. Therefore, again, breaking down the proteins can definitely affect the microbe that we want to either kill or we want to inhibit its growth. So things like alcohol, moist heat, so high temperatures with moisture, okay, kind of like humidity that can affect and denature the proteins. Now, what this table is showing you, it's showing you that these physical and chemical, methods that we use to go ahead and either kill or basically inhibit the growth of certain microbes, this table is showing you how some of these microbes can actually be pretty resistant. Okay, so we're kind of seeing, which microbes are more resistant compared to others. So let's see the least resistant. The least resistant are enveloped viruses. Those would be, easily targeted, okay, and they're not as resistant, for example, compared to prions. Prions, as you can see, they are the most resistant. Prions are pretty hard, to get rid of. Now, first of all, you might be wondering, what are prions in the first place? Well, prions are basically misfolded proteins. And so the reason why it's kind of hard for us to get rid of prions, we kind of just went over the list, right? We said that a lot of these physical and chemical methods that we use target things like a cell wall. Prions don't have a cell wall. Things like a plasma membrane. Again, they don't have any of that. They don't have, nucleic acids. They're basically just misfolded proteins. And so that's why it's kind of, hard to get rid of them. Now we're gonna, talk about things that can kill a prion, those misfolded proteins, and it's usually like, super high heat for a long time can go ahead and kill a prion, but they are the most resistant, followed by endospores, those bacteria endospores, because of their hard outer shell. Again, makes endospores pretty resistant, followed by Mycobacterium, and Mycobacterium are acid-fast bacteria, so they have this lipid waxy layer, which again kind of protects them, okay, from many agents, and so therefore they are again pretty resistant, followed by the genre like Staphylococcus and Pseudomonas, and under those two genera there are, Staphylococcus species that are pathogenic and Pseudomonas species that are pathogenic, and so again they're kind of resistant as well. But as you guys can see, that the resistance level is starting to kind of like lower, then followed by the protozoan cysts. Those are kind of like the dormant, inactive stage, and then the trophozoite, which are like the growth stage of those parasites, followed by most gram-negative bacteria. Fungi and fungal spores would be less resistant, then followed by non-enveloped viruses. Most gram-positive bacteria, as you guys can see, like if you compare gram-positive bacteria to the gram-negative bacteria, you can see that most gram-positives, they are less resistant to gram-negative, and the reason why gram-positive, they're less resistant to gram-negative because the gram-negative have that outer membrane. And then the least resistant would be the enveloped viruses. Now, what this table is trying to show you right here, again, before we go into the details of the different physical and chemical methods of destroying these microbes, this table is specifically talking about endospores versus vegetative cells. And this table is trying to show you that endospores, they are way more resistant to vegetative cells. Endospores, as we know, bacteria produce endospores when the bacteria is either old or when the bacteria is running out of nutrients. The bacteria goes ahead and produces these endospores, those kind of like dormant-like spores. And those endospores, they are meant to go ahead and withstand pretty harsh conditions. And the outer shell of the endospore is extremely tough, right? So that it can go ahead and withstand these extreme harsh conditions. And so getting rid of an endospore is way harder compared to getting rid of the vegetative cell. The vegetative cell is basically the normal growing cell, like the normal growing bacteria that is basically forming the endospore. So this table is just showing you that whether you're using like moist heat. What's an example, by the way, of moist heat? Like when you boil something to disinfect it, or when you use an autoclave. Those are kind of examples of moist heat. It shows you that to destroy an endospore, you need to go ahead and crank up that temperature like pretty high to destroy the endospore, versus if you just want to, kill the vegetative form of the bacteria, you can just put it at ATC compared to 120C. So it kind of shows you that in this case, the endospores are like 1.5 times more resistant compared to the vegetative cell. And you can see that this is true for all the other methods, right? Whether you're using radiation or some, sterilizing gas or, a sporcidal liquid, you can see that again, you will have to use more radiation to kill an endospore. You would have to use a higher dosage of that sterilizing gas to kill the endospore. You would have to expose the endospore, for example, to that harsh chemical for a longer time, right? See how right here you have to leave that chemical on the endospore for 3 hours compared to only 10 minutes if you want to kill the vegetative form. And so you can see that in this case, the endospore is 18 times more resistant. So again, this table kind of shows you how more resistant the endospores are compared to their compared to the vegetative form of the bacteria. Let's talk about a few terms. The first one we're going to discuss is one that's known as sterilization. Sterilization is actually the most extreme protocol used to control microbial growth. Sterilization is basically the complete removal and the complete removal or basically killing of all vegetative cells, viruses, endospores. So it's the complete killing and removal of the microbes. Now, sterilization protocols are usually used in lab settings, medical, healthcare, basically, clinics or hospitals, even food industry settings, because some items, it is extremely important that certain items should be completely free from all microbes. And so sterilization can actually be accomplished, for example, through exposure to high heat, high moist heat, such as when certain items are placed in an autoclave machine. Or it can be accomplished through filtration. Of course, an appropriate filter has to be used, or through certain chemicals, pretty harsh chemicals that can go ahead, as I said, and kill basically all the microbes, viruses, including endospores. And so therefore, when we are using the term sterile, if an object is sterile, we can't say, oh, this object is somewhat sterile or slightly sterile. There's no such thing. Either the object is sterile or not. And if the object is sterile, that means there are 0 microbes. Like all the microbes are now, dead. Of course, we cannot sterilize our body, or you can't sterilize your skin, because of course our human tissue would not be able to withstand such harsh chemical agents that are used to sterilize objects. And so therefore, again, when we clean when we clean our skin, we are not sterilizing our skin. On the other hand, what we are doing is we are degerming our skin. So we'll talk about that in just a minute. And so as I said, sterilization is also used in the food industry. especially, for example, when they are, when they are prepping like canned food. Now, cans are going to be, the environment inside of a canned food is going to be anaerobic, and certain bacteria can withstand anaerobic environments. And if these bacteria are capable of producing endospores, now these endospores would actually be able to go ahead and become viable in anaerobic environment. And so therefore these food industries, they would have to ensure that they go ahead and they actually heat up, heat up the food items to high, to high degrees, like to 121 degrees Celsius, or even higher, and again for a minimum period of time, just so that they can ensure that they are killing again the microbes and reducing and killing, like, the endospores. Just to ensure that again no one is going to get sick when they go ahead and consume the canned food item. And so therefore, when you go ahead and before you consume, like, canned food, kind of take a look at the can. If it looks bulgy, okay, especially like at the top of the lid, usually that's not a good sign, right? Because it could mean that there are bacteria in there, and the bacteria is growing and it's kind of producing those gases, and that's why the lid part is bulging out. So that's kind of something to watch out for. Now there's another term, and that is disinfection. Now this infection, it's meant to destroy the vegetative cells, but it cannot destroy the endospores. And so usually this infection is basically going to inactivate most of the microbes on the surface. Now, again, it's not going to be killing all of the microbes. So with the disinfecting surfaces, you're still going to actually have some microbes that remain on the surface. And so therefore, a disinfectant item is not considered sterile since there are still microbes on it. Usually disinfectants are, used more often because they are, easier to prepare. They're less expensive, so they're kind of easy also to use. There are natural disinfectants that can be used, such as vinegar, because it's highly acidic, so it can go ahead and kill a lot of microbes. Now, of course, there are, chemical disinfectants like bleach and iodine. There are also physical methods that can be used to disinfect items, and that would be to go ahead and boil certain items before using it. Now, disinfectants, again, usually because of their high concentrations, etc., they are going to be considered harmful to the human tissue. So again, you wouldn't want to disinfect your skin, for example, by pouring bleach on it. You will definitely burn yourself. Now, there is another term, and that is decontamination, or you can refer to it as sanitization. Okay, and that's basically where the microbes are, being removed, and the idea here is to try to basically reduce the contamination levels to safe. So again, it's not like you are removing again all the microbes, but with decontamination, what we're trying to do is basically reduce the number of the microbes so that we can reduce the contamination, risk. Things like soaps and detergents, etc., are used. So again, decontamination or sanitization processes are usually used at homes, at restaurants, etc., to cleanse and sanitize the cooking utensils and the dishes, etc. And the last one that we are going to go ahead and talk about is antisepsis or degerming. So antisepsis, that's basically you know, for example, when you go ahead and scrub your skin, especially if you are working in the healthcare field, you know, especially before a surgery, the medical, you know, doctors, nurses, etc., would actually have to go ahead and scrub their skin before, you know, before starting the procedure, because you don't want to go ahead and expose your patient to all the microbes that you have on your hands. And so therefore the antisepsis is basically you are reducing the number of microbes on the human skin. Again, you are not removing all, you know, you're not removing all the microbes on the human skin. You are just reducing the number of the microbes on the human skin. And so again, our human skin is not sterile and we cannot sterilize it. So with de-germing, you're basically trying to reduce the microbial load, for example, on the skin or on your tissue. And again, this is done usually by, you know, gentle, like, you know, to firm scrubbing. Hand washing, you can use things like soap and alcohol, of course. And again, it's just meant to reduce the microbial load. And so we're like, you know, things like alcohol could be used, things like hydrogen peroxide, iodine. Again, those are, you know, common agents that are used with the antisepsis. So let's go ahead and kind of move on. Now, this chart right here, what it's trying to show you is the different, you know, physical agents, chemical agents, or mechanical, you know, removal methods. So one of the physical agents that we use is basically heat or radiation. Now, with heat, we actually have, you know, two types of heat that we are going to discuss, dry heat or moist heat. Examples of dry heat would be to incinerate, and that's basically what you do in micro labs when you go ahead and take your loops and basically pass it, you know, into the flame until it turns red. And basically, if you have bacteria on your loop and you go ahead and stick it in the flame, basically the bacteria is going to sizzle and it's going to burn right away. So that's considered incineration, or, you know, the dry oven. And these are basically actually sterilization steps, meaning like, again, all the bacteria are going to die, including the endospores, etc. Moist heat, an example of moist heat would be, you know, when you're boiling water, you know, and let's say you're sticking items in the boiling water to go ahead and help, you know, disinfect it. Pasteurization process, that's another, you know, process to help, again, reduce the microbial load, for example, in milk. Right, so when we say the milk is pasteurized, again, we're doing this to reduce the microbial load in the milk. Milk is not sterile, right? So it's not sterile because it is still going to contain, you know, microbes. But again, what we're doing with the pasteurization process is we're reducing the microbial load to basically safe levels. Or, you know, steam using, like, you know, high pressure. This is going to sterilize and it's going to kill, you know, everything, including the endospores. With disinfection, as I said, you're not killing the endospores. Radiation is another physical agent that we use. Now we can use ionizing radiation and or non-ionizing. Example of ionizing radiation would be things like when you use X-rays or gamma rays, and those are basically pretty, you know, pretty tough. Like they can pass through objects, okay, through plastic, etc. And so those would actually be able to sterilize and again kill all the cells, including you know, including the endospores. And so as you can see, like, you know, sterilization, you know, when you incinerate things, when you put it in dry oven, when you put it in, you know, steam with high pressure or expose it to such, like, you know, harmful rays, all of these are going to have to be, done basically or used on inanimate objects, right? Our human tissue would not be able to withstand withstand sterilization. Now, UV rays, by the way, are less, they penetrate less compared to these, rays right here. So UV rays can go ahead and they're basically going to be under the group of like disinfecting, where you are destroying or removing the vegetative pathogens, but it's not going to be able to kill the endospores. And again, it's used on inanimate objects. Now, when we move on to chemical agents, we could use certain gases, we could use like, certain liquids. Now, certain liquids can be used on our human tissue, right? And that would be like antisepsis. So again, when you go ahead and pour some alcohol or, clean your skin using, let's say, for example, hydrogen peroxide, now that would be antisepsis. And then, of course, there are liquids that we should not at all, be pouring on our skin because they are pretty harsh, so they could actually be under the group of like disinfecting, killing vegetative cells, or sterilization, like killing everything, including, of course, endospores. Now, mechanical methods, for example, filtration, right? So when you filter the air or, when you filter the liquids, liquid filtration is going to actually result in sterile, sterile product where, again, all vegetative cells, including endospores, are going to be killed. Air filtration is usually going to, decontaminate. So this is where, again, you are removing most of the microbes, but it's still going to, you're still going to end up with some microbes. Now let's take a look and see the difference between moist heat and dry heat. Now, moist heat, it's usually like, set at lower temperatures. Exposure time is not, not for a very long time, as you can see, 3 minutes, 10 minutes, 15 minutes. Now, of course, the time is going to vary based on the temperature. As you can see, the higher the temperature, right? So the higher the temperature, the less time it's going to take to sterilize the object. And the way it works, moist heat, is basically it kills these microbes by basically coagulating, kind of like clumping and denaturing, like breaking down the proteins of these microbes. Example of moist heat is, the autoclave, or basically, boiling water, or the pasteurization. Those would be examples of moist heat methods. Now, dry heat is usually going to require longer exposure, okay? So as you can see, time-wise, you would have to expose the object for a longer, period of time. And what it usually does is it goes ahead and dehydrates the bacteria, and it denatures its proteins. What's an example of dry heat? For example, when you use when you use the open flame in the microbiology labs to go ahead and sterilize your tools that you are using, that's considered, dry heat. Hot air oven, that's also going to be considered a dry heat. Okay. Now, as you can see with dry heat, as I said, you go ahead, it requires like a higher period of time, and temperature-wise, as you can see, the higher the temperature, right, the less exposure time though, okay? So kind of like moist heat. The higher the temperature, the less time you're going to have to expose the microbe, okay? So that you can go ahead and kill it and basically achieve sterilization. Now let's go ahead and take a look at a few examples of moist heat methods. And so one method is the boiling, boiling water. And so, boiling water, you can accomplish that at home, right? So we usually like boil the water to like 100 degrees Celsius. And now, this is not going to kill all microbes, remember, because how some microbes can actually withstand 100 degrees Celsius. So again, this method is going to be to disinfect and not to sterilize. Why is it not, for sterilization? Because again, this method is not going to be able to kill all cells, or endospores, and so therefore it's a disinfection method, not sterilization. And you can usually like expose the material, for at least like 30 minutes so that you can at least, kill most of the, or a good number of the microbes. Now, the thing is though, once you've, let's say, placed an item in the hot, boiling water, and then take it out, well, I mean, once you take it out, this item can be easily recontaminated, okay? Again, it's usually used like, at home for, for disinfecting, bottles, utensils, etc. Now, another example of a moist heat would be, for example, the process of pasteurization. And so things like, milk and juice and beer and wine, they are pasteurized. Now, these items are not going to be sterile, okay? Milk, beer, etc. They do have microbes. Um, but again, the pasteurization, it's basically a method where heat is going to be applied to these liquids. And basically what we're trying to do is, reduce the number of the microbial load, to safe levels and, to kind of prevent quick spoilage, okay? And by the way, that's why milk, even though, you haven't opened it or anything, it can actually go bad because the milk is not sterile. It does have microbes. So even if you've left it in the fridge and you didn't open it, eventually it is going to go bad. Um, and so again, this is not a sterilization process, and so these objects are not going to be sterile because it does have microbes. For example, milk, as I said, it's not sterile and it can actually contain, milk can actually contain like 20,000 microbes per milliliter. And again, this is kind of like as long as it's within the normal range, then it is going to be safe for us to go ahead and consume. Now, another method, and that would be um, steam, basically under pressure now, or basically autoclaving. Now, this one is a sterilization process. This one is going to kill all the microbes, including endospores. So this one, it is going to be able to sterilize, the objects. And so again, we usually, in microbiology labs, what we usually do is we go ahead and use these autoclave machines to basically sterilize all of the, plates that you've been using, the glass pipettes that you've been using, etc. So you go ahead and kind of put it in this like, chamber and these autoclave machines are usually set to like, 121 degrees Celsius. And now for how long do we autoclave certain items? It varies. So you can set the autoclave machine, for like 10 minutes if you didn't put a whole lot of stuff, or you can set it for like an hour, okay? And that's again if you've placed like a whole lot of stuff in the autoclave machine. And so Um, things like glassware, okay, metallic instruments, the plates that we use in the lab, the glass pipettes, etc. Those items can go ahead and be placed in an autoclave machine. Now, even though you might be wondering, well, aren't the plates, that we use in the lab, aren't they made out of plastic? So how come we can place it in an autoclave? Well, because usually those petri dishes we don't reuse, okay? So yes, they're gonna be, kind of destroyed, but it's fine because all we want to do right now is just kill whatever is on your plate because we do go ahead and discard those plates. We don't reuse them. Um, and so items though that have like wax or substances that have like wax or oil, etc. Or powders, those we don't usually stick in the autoclave machine. We go ahead and sterilize them using different methods. Dry heat, as I said, an example of dry heat is incineration. For example, when you go ahead and stick your loop right in the flame, and basically, that open flame or that Bunsen burner is going to kill the bacteria right there on the spot. Um, and so we go ahead and sterilize, all sorts of stuff that we use. So this is a sterilization process. So incineration is a sterilization process because it is going to kill the microbes right away for you, and it's going to kill all of them. And so basically, things that we go ahead and sterilize in the microbiology lab, things like inoculating loops, needles, etc. Um, Now, they usually have, large actually incinerators that are found in hospitals and in research labs because sometimes when we want to get rid of like a huge bulk, okay, of infectious material, they go ahead and they put it in that large incinerator. Now, another example of dry heat would be the hot air oven, and usually it's applied, when they want to sterilize. Again, it is a sterilization process. So when they want to sterilize things like glassware or metallic, instruments, or actually powders and oils, they can go ahead and use the hot air oven, which is basically it can be actually run either by gas or electric, and it radiates high heat. Again, it's kind of like you place your items within this compartment right here, and then really hot air circulates in in the chamber, and sterilization usually requires you to go ahead and set this hot air oven to like 150 degrees Celsius or 180 degrees Celsius, and you would leave it to run for like 2 to 4 hours. Again, it depends on how many objects you're placing in that hot air oven, etc. But that's again, those two processes would be they would be able to achieve sterilization. The object would be completely sterile after going through those methods. Um, Now we're going to talk about radiation. So radiation is basically another, it's another physical method that we use to go ahead and disinfect, surfaces. And there are multiple actually, radiation that we can use, things like gamma rays, X-rays, or UV rays. Um, Now, gamma rays and X-rays, they are much actually stronger than UV rays, and they can actually penetrate through, through several different types of surfaces. UV rays, by the way, are considered non-ionizing radiation. Ionizing radiation would be the gamma rays and the X-rays. Um, Now, the gamma rays and X-rays, they usually go ahead and use it, and it is going to be a sterilization process because these ionizing radiation rays are pretty strong, so the object will become sterile. So they go ahead and kind of use it to sterilize things like drugs or vaccines or basically, certain medical instruments like syringes, etc. They can even use it to go ahead and sterilize surgical gloves. So again, the gloves, by the way, that are used during surgery are not like regular gloves that you can buy off the shelf. No, these gloves must be sterile. So even gloves have to be sterilized before using them, and in especially like during surgeries. um, and then, of course, like all sorts of like medical tools, can also be sterilized using X-rays and gamma rays. Um, Now, we're also going to talk about, let's see, we're also going to talk about non-ionizing radiation. And so non-ionizing radiation, an example would be UV rays. Um, Now, your UV radiation is not really as penetrating as gamma rays and X-rays. And so therefore, UV radiation, even though it does pass easily through air, um, kind of less through liquid, and it doesn't, it's very poorly, through solids. So UV rays doesn't do very well at all if it's trying to penetrate through solids. And so therefore, if you want to disinfect an object, therefore, by the way, UV rays are not going to they're not going to lead to a sterile object. Okay, versus gamma rays and X-rays, they can't. But UV rays, at the end of the day, the object is not going to be sterile. It's still going to have microbes on it. And that's because, again, the UV radiation doesn't really pass through solids very well. And so the object that you want to disinfect using UV rays will actually have to be directly exposed, okay, directly exposed to the UV, to the UV rays. Um, Now they actually, have certain kind of like, UV machines that they go ahead and kind of place in a hospital room, and basically the ultraviolet light is going to go ahead and disinfect the air, disinfect the surroundings. Um, and so, again, UV rays can be used, to destroy things like fungal cells and spores and bacteria, vegetative cells and viruses. Now, of course, if you're trying to kill the bacteria spores are way more resistant, compared to like the vegetative cells. But the bacteria spores can be killed by UV rays. It's just you have to increase, you would have to increase the time of exposure, okay? And as I said, like directly place it, in direct contact with the UV, with the UV rays. But still, the UV rays are not going to be able to sterilize the objects. So you're still actually going to have some microbes. Um, and so again, gamma rays and X-rays, those would be considered the ionizing radiation. UV rays would be considered the non-ionizing radiation. And you can see now they have kind of like, these machines right there, they sent into, hospital rooms, and then the UV rays basically tries to kind of, zap like, all of the surroundings. And again, it's going to reduce the microbial load on these surfaces, but again, it's not going to make these surfaces sterile. Okay. Now, how do UV rays, by the way, kind of like, destroy bacteria? What they do, let's say this is a normal segment of DNA within the bacteria, so you can see the bases linked together. What UV rays are going to do is they're going to result in something called thymine dimers. Do you see how right here, those two thymine bases, they basically go ahead and connect with each other. And this is basically going to result in that part not, that part now of the DNA, because it's linked like this, which is not normal. It's going to result in that part not being able to get replicated properly or transcribed or translated properly, and so therefore it is going to affect, right, the production of whatever that part of DNA is encoding for. So again, this is what UV rays would do. They would result in these thymine dimers. Now, one thing to note is when we place our food in the fridge, right? One thing to note is that cool temperatures are not meant to kill the bacteria. Cool temperatures are meant to slow down the growth of the microbes in the food. And so therefore, we actually consider cool temperatures to be considered microbe static and not microbicidal, because they're not going to kill the bacteria, but what they're going to do is they're going to try to, slow down its growth rate so that you can preserve basically the food items, for a period of time so that they won't go bad, pretty quickly. Um, and that's why you hear of certain certain microbes, again, not being affected by the cool temperatures, for example, like psychrophiles, right? They enjoy the the fridge temperatures. And I mean, you hear of certain microbes or like, endospores that are actually that can withstand freezing, freezing temperatures. We even use freezing temperatures when we want to even preserve the cultures of bacteria. So in a lab, when we want to preserve a certain culture of bacteria for like months or years, what we do is we go ahead and we freeze it. And so, we know that by doing that, we are not killing the bacteria. All what we're doing is we're just really preserving it by slowing it down. And if we want to use it later, we can go ahead, kind of thaw it out and use it. And so, therefore, cooler temperatures are definitely microbe static, because they're just going to go ahead and slow down the growth of these cultures. And as I said, there are even certain pathogens that are capable of surviving. Even like, they were able to survive in a fridge for several months, such as Staphylococcus aureus, such as certain species of Clostridium, certain species of Streptococcus, and some of these species are actually pathogenic. So if the food is contaminated with these species and we go ahead and consume it, it can definitely make us sick. Um, so let's go ahead and move on. So next we're going to talk about filtration. So filtration is basically a physical method that's used. It can achieve sterilization, especially liquid filtration. So liquid filtration can achieve sterilization. And they usually use it, for heat sensitive materials such as vaccines and antibiotics. So vaccines and antibiotics, if you want to go ahead and sterilize them, we can't really stick them, for example, in an autoclave machine because they're not going to be able to withstand such high temperatures. But at the same time, we do want to go ahead and sterilize that vaccine before, before injecting it into the arms of individuals. And so one way to go ahead and make sure that this vaccine is completely sterile, or again, everything is killed, is to go through that liquid that liquid filtration. So filtration is a pretty efficient, method to go ahead and remove and basically kill bacteria. And as I said, it can make the liquid, let's say that you are preparing, let's say you are preparing a batch of vaccines or a batch of like antibiotics, filtration is going to make sure that these are going to become sterile. Let's talk about a few chemical agents that are used. These chemical agents that are listed right here for you, halogens such as chlorine and iodine, some oxidizing agents such as hydrogen peroxide, some aldehydes, and some gas sterilants. All of these are actually going to achieve the sterilization process because they're going to kill all different types of microbes, including viruses and endospores. So all of these are actually going to, achieve that the object that's getting sterilized is indeed, sterile because all the microbes are going to be killed, as I said, including the endospores. Now, halogens, those are basically chemicals that include things like chlorine, iodine, and fluorine. So we're going to talk about chlorine. Now, chlorine can kill all, microbes, including endospores. So as I said, sterilization can be achieved. Now, chlorine, usually when it gets mixed with water, it releases hypochlorous acid, and so therefore individuals that tend to use a lot of chlorine, they got to be careful, right, because of that acid that's going to be released. That acid enters the cells pretty quickly, and what it does, it can denature the proteins, denature enzymes within the microbial cells, and it can also suspend metabolic reactions. And so chlorine is used, to disinfect, you know, uh, multiple things, multiple instruments. They use it also in the, sterilize and food equipment, for example. It's used, in restaurants. We even add chlorine, right, to the pools. So you add like chlorine tablets to go ahead and clean the pools and spas, etc. Um, Now, another type of a halogen is iodine, and iodine can also kill endospores and microbes, and again, sterilization will be achieved. Chlorine interferes with the metabolic activities of the microbial cells, and kind of messes up with the hydrogen bonding and disulfide bonding of the proteins. And so again, basically messes up with the proteins, bonding, so it will mess up with its structure. Some iodine can actually be mixed in with some other, with some other like fluids, so that it can be used. And we do use iodine to go ahead and prep the skin for surgery. So before, like, individuals would have in surgery, they would usually give patients kind of like a solution, and the solution has iodine plus some other, some other stuff like mixed in together. And they would tell you, okay, before the day of your surgery, go ahead, take a shower and apply this solution, especially over, the skin area that they're going to go ahead and cut open. Now there's another, type of chemical known as oxidizing agents. An example is hydrogen peroxide. Now there are other agents out there, but hydrogen peroxide is one of them. Now the way hydrogen peroxide works is it goes ahead and produces free radicals, and those free radicals are highly, highly toxic to the, to the cellular components of the microbes. um, so um, this is an example, as I said, of an oxidizing agent, hydrogen peroxide. Well, now we do use hydrogen peroxide, not of course in such high percentage, when we go ahead and apply it to our skin and to our wounds, okay? So again, hydrogen peroxide can be used, on skin, like, to cleanse wounds. Some people even use it as a mouthwash. Um, they can even apply it, to someone who has bed sores. Um, Now, aldehydes is another group, and aldehydes can go ahead and kill, microbes and endospores, etc. And the way aldehydes work is they go ahead and disrupt the activity of proteins, so the proteins will not function properly. A lot of these, by the way, chemicals that we are using could actually be considered broad spectrum. Broad spectrum meaning that um, they can kill both gram positives and gram negatives, or they can affect basically both gram positives and gram negatives microbial growth. That's what it means by broad spectrum. Now, gaseous sterilants or disinfectants, an example would be, ethylene oxide. Those, they basically react with the DNA of the microbe and the proteins, and they can basically block the DNA from being replicated properly. And so this is kind of how they act. They do use it, to disinfect things like plastic materials and syringes and, things like that. So medical, basically, supplies, surgical supplies, etc. Um, Other groups, they are called phenols. So phenols, now those products, phenols, alcohol, certain detergents, certain heavy metal compounds, basically this whole entire list right here, they're not going to achieve sterilization because they're not going to be able to kill all microbes. And not, they're not also going to kill endospores, and so therefore they are not considered, to achieve sterilization. Okay, so one group is phenols, and those can kill some bacteria, some viruses, some fungi. Again, not all. So therefore, again, you're not going to sterilize the object if you use phenols. Now, phenols, go ahead and we usually use them in, high concentrations, and they basically disrupt the cell wall of the microbe and the cell membrane, and they can even disrupt like the proteins. But for them to do a good job, like you would need to use like high concentrations of these phenols. Now, phenols, sometimes they add it into the drains, okay? This is kind of like, one common place that they would go ahead and use, use phenols. But as I said, you would have to use such high concentrations, and so a lot of them are, pretty toxic. And so again, whoever is handling phenols, they have to be kind of careful handling the phenol. And actually, like a lot of phenols out there, they got banned. So they were banned, so they're not supposed to use them, okay? Let's see. There is chloric hexadiene. Again, it can target specific, bacteria, some viruses, some fungi, and they target again the cell membrane. So they kind of, break the cell membrane, also the cell wall. Also, like, they can denature proteins. Um, they're less toxic. Okay, so they're less toxic compared to phenols. Um, they can actually use it, in components that are added, to hand scrubbing, components that they use, like so when they hand scrub before going into surgery, or, sometimes they add it when they're prepping the skin for surgery. Again, once the patient goes into the surgery room, before they cut them open, they still have to prep the skin. They still have to kind of, clean it up. Alcohol is also going to target, again, most bacteria, viruses, and fungi. Um, Now, with alcohols, concentrations of like 50% and higher can actually dissolve the membrane of the microbes. And so, therefore, you would notice that when you are using, like, home, like, alcohol, like the ethanol bottles that we purchase, you will see that it has like 70%, and some of them have higher, because again, like higher concentrations would be better to disrupt the cell membrane of these microbes. Um, Now, alcohols tend to evaporate, pretty fast. Okay, so because they evaporate pretty fast, their effectiveness, doesn't last, for a long time. And again, though, we have to kind of make sure that when you are using alcohol, like, I mean, you're not overusing it. Because again, you don't want to inhale all of these vapors. It can definitely affect you. Um, Detergents, okay, so detergents, definitely, like, when you're, washing clothes, or like, you're using certain detergents to go ahead and kind of clean, restaurant utensils, etc. Um, some of these detergents are known as quaternary ammonium, or we basically refer to them as quats. And the way they work is they go ahead and disrupt the plasma membrane of the microbes, and that can cause, the microbes definitely to die when their cell membrane is affected. Um, some of these products, they are not broad spectrum, meaning, so again, broad spectrum would mean that this agent can go ahead and affect, both gram positives and gram negatives. Um, those quats are actually considered narrow spectrum. Narrow spectrum meaning that they can target like only either gram positives or gram negatives. So the quats, they are known to be more effective against gram positive bacteria. Okay. Um, Heavy metals, okay, heavy metals compounds, so it would include things like mercury, silver, things like that. I mean, it could include also like other metals. Those are basically, you can mix them in, by the way, with like some other stuff. You can mix them in aqueous solutions, or even like, in some alcohol, solution. Um, these are, pretty good, effective antiseptics, and they are used basically as actually preservatives. So they would add them in cosmetic products. They would add them in certain ophthalmic solutions, solutions that you would put in your eyes. Um, and so some of them are used as, like, topical ointments. Okay, so you would put it as an ointment, like, on your skin. Now, some microbes can actually start developing resistance to these, metal products. Um, those metal products are not going to achieve sterilization because they are not effective against endospores and they cannot kill endospores. Um, And then this last table right here, you guys, shows some of the, some of the agents that are used, and what type of like bacteria, they would be highly effective against. So chlorine, could be used against Mycobacterium tuberculosis, things like hepatitis A virus. Um, Alcohol would be good against Staphylococcus aureus, E. coli, etc. And it kind of shows you that at least, for the alcohol, you want to get something at least at 70%. So again, when you guys are purchasing alcohol, you want something that says like 70% or higher, okay? Because I mean, this would be a good percentage to, to kind of kill such microbes. Um, Hydrogen peroxide, at least 3% of the hydrogen peroxide would be needed, to kill things like Staphylococcus aureus, Neisseria, gonorrhea, the herpes virus. And as you can see, though, depending on the strain or depending on the bacteria, sometimes the timing is going to differ. So how much time is it going to take to kill those bacteria? As you can see, like Neisseria gonorrhea is definitely more, it's definitely less resistant, right, compared to, for example, Staphylococcus aureus. And then these are the quats, Staphylococcus aureus, take 10 minutes to go ahead and kill. Salmonella, typhi, again, 10 minutes. And then ethylene gas, again, it can be effective actually against the flu virus and, about 25 hours and kind of like, such a pretty high concentration though, so that it can kill, that virus.